Hi, I'm Angelique Regnier-Golanov. I'm an instructor in the Department of Neurosurgery. I work in the Laboratory of Cerebrovascular Research, and this is my great pleasure to introduce you today to our latest work that we conduct in the lab. So today I'm going to talk to you about the Post-SAH syndrome, which is a cognitive behavior in mice model following SAH. My interest and my goal is to develop new therapeutic approaches for the treatment of long-term consequences of subarachnoid hemorrhage using clinically relevant animal model. My background is I am a neuroscientist. I trained at the University of Paris, where I obtained my PhD thesis. I conducted research on the neuroinflammation and remote associated damage in an interesting model of temporal lobe epilepsy. I continued my training at the Baylor College of Medicine with a postdoctoral fellow, where I studied the main comorbidity of temporal lobe epilepsy that are refractory to treatment, which is learning and memory. And this unique background made me particularly qualified to conduct research in the post-SAH syndrome following subarachnoid hemorrhage. Today I'm going to introduce to you a paper that was recently published in our laboratory. So the main focus of our lab is subarachnoid hemorrhage. Subarachnoid hemorrhage is a bleeding in the subarachnoid space, where subarachnoid hemorrhage is a non-traumatic stroke, is the least frequent of stroke, but is fatal with 50% of fatality after the bleeding. However, SAH survivors, up to 95% of them present cognitive deficit. It touches a population that is rather young, as the rupture of aneurysm occurs usually around 55 years old. And this rather young population, which represents about 2 to 3/3 of the population, cannot go back to work. So we are using a relevant model of subarachnoid hemorrhage, which is called endovascular perforation of the circle of Willis. As we can see here, we induce bleeding at the base of the cranium by introducing a microfilament through a stump that we created to the external carotid artery, and that we conduct all the way to the circle of Willis, where we perforate the vessel to induce the bleeding. Our model is particularly relevant to the clinic, as most of subarachnoid hemorrhage are occurring at the base of the skull and are the result of rupture of aneurysm at the base of the skull. SAH patients are surviving better the initial insults. However, they experience strong cognitive impairment that are actually not being treated in clinic. We used our model to establish a battery of 5 well-established behavioral tests to show our cognitive impairment and to provide this way a technical platform to test the treatment. So we started to test our animal 4 days following SAH in the open field test. The open field test is a well-established test in the neuropsychiatric disease and in the epilepsy community. It tests the propensity for animals to explore new open areas, and it measures open field measure the test for the anxiety and for the locomotion. The anxiety is measured by the percentage of time that the animals are spent in the center, like we can see here in yellow in the box. And we can see in our animals at 4 days, that 4 days post-surgery, there is no difference between SAH and sham. However, when we tested the animal 30 days after, in a chronic window, 30 days after the SAH, we see that our SAH animals spend less time in the center, showing that they present early sign of anxiety, and they will have a tendency to spend more time around the walls, like we see in this representative illustration, where there is a higher tracking red path closest to the hole, which is called sigmostasis, and it's an indicator of anxiety. This test is also used in the community to check for the locomotion. We see that 4 days after SAH, our animals are presented less locomotion than 30 days. However, they recover at 30 days. So we can safely say that our anxiety indexes is checked because of it's not because of a reduced locomotion. By this test, we saw that our model is relevant to the clinic, as it was indicated that up to 30% of the SAH survivors developed persistent anxiety. Another test conducted at 4 days is a social interaction, or testing the sociability of the patient. Social interaction is known to involve it's a complex task involving several brain areas. So this is also a way to check for the overall well-being. Our experimental mice were put in an area with an intruder mouse that is the same age, the same sex, and the same strain. And they are placed with each other during 5 minutes, and the 4 following behavior will be recorded to measure for their social interactions. So in the rodents, they are interacting by facial sniffing, orogenital sniffing, follow or pursuit behavior, as well as inactive avoidance. We see in our experimental mice, when we sum up all the time spent in this behavior, that the SAH mice spend less time interacting. Interestingly, we also measure this behavior in our intruder mice, and we see that they spend themselves less time with the SAH, showing that a normal conspecific, or at least a healthy conspecific, will have a tendency to recognize their mate to detect if there is presence of disease or not. The last test was conducted in 4 days post-SAH. It's a Y-maze task, or spontaneous alternation. The rodents are known have a propensity to explore the novelty. So they are placed in an arena called the Y-maze, which has 3 arms, and they will subsequently visit all the arms that they don't know. And when they missed, so they will go successively after arm B, C, and D. This task is to measure the working memory. It's also called the episodic memory, which is essentially the memory that we need to accomplish everyday tasks. We have to remember what we were just doing a few seconds ago. It's measured by the percentage of alternation. We measure the number of successive visits that they conducted in each and every arm, divided by the total visit, which gives us the number of errors of if they alternated properly. We see that in the SAH mice, they still alternate less than our sham, showing that a slight deficit in their working memory. We control for the total distance and the total entry to see that there was no difference in the overall traveled distance in this Y-maze, to show that our indexes of cognition is due to the impairment of cognition and not to trouble in locomotion. We conducted our study in a subchronic time window, 30 days post-SAH, in the Barnes maze. The Barnes maze is a task to assess spatial memory. The animals are put in a platform where they are about 1 meter above the floor. There is 20 holes drilled into this target, and among one of these holes there is a hidden platform, a hidden gold box. The platform is brightly lit that acts as an aversive stimulus for the mice that will have a tendency to find this gold box to escape the lights. We measure the latency, the path length, and the total error, and we see that there was significant difference in the latency and the total error between the SAH mice and the sham mice, showing that starting after the trial number 6, so initially the SAH mice take more time to find the gold box, or they present more error to find the gold box, but eventually after the trial number 6, they present the same pattern as the SAH, as the sham mice, seeing that they eventually learn. We also recorded the strategies from 1 to 3 to see from the less complex to the more complex, to show what strategy they use and if they change among trials. And we see that there is no difference between the sham and the SAH mice, but they used faster strategy, both groups, according to time. After third day of training, on the first day we conduct a prod test which is supposed to measure the time. We let them 90 seconds in the platform for them to explore freely where the gold box have been removed. We see that SAH and sham mice spend the same amount of time in the target 24 hours after the last trial. We also see that the SAH mice have a bit longer time to find the appropriate gold box. And both mice conducted the same error. So this test shows that there is impairment in the spatial memory. However, they recover they recover pretty quickly. Interestingly, recent studies showed in a virtual maze that they detected spatial memory deficit in SAH patients, again illustrating the relevance of our clinical model. The last test we tried, 30 days post-SAH, is the novel object recognition. The animals are habituated in the arena 2 days before taking the test, where they are placed twice 10 minutes per day to explore the arena, that the taking place of the context isn't going to influence the test. On the day of the test, the animals are placed during 5 minutes in the arena with 2 identical objects that they are at free leisure to explore for 5 minutes. They are put back in their home cage, and 2 hours later, they are being tested, and one of the objects is removed. And we will measure the time they spend to explore both objects, the object A that they have been exposed to, that is a similar object, and the object B that is a novel object. We see after counting that after summing up the time explored, that the SAH mice spend more time exploring the novel object, while the SAH mice don't make any difference. We also illustrated this with our novelty preference index, where we see that there is a higher index for the sham mice. This test also put back into clinic or in human use, we see that there is a deficiency in emotion recognition in clinical studies following SAH, and this impairs their ability to return to work. So the conclusion of our work is that limited subarachnoid hemorrhage, as we produce a limited bleeding at the base of the skull, induces long-lasting behavioral abnormalities. We identified an anxiety-like behavior that developed at 30 days. We identified a significant decrease in social interactions and in working memories. We also showed a slowing down of spatial memory, even if the animals were able to catch up at the end. And we saw also evidence for a significant decrease in recognition memory. So with this current work that just have been published, we have developed a platform with an animal model and identification and validation of a battery of behavioral tests that mimics human pathology. This platform will be used for translational studies looking for therapeutic approaches and therapeutic treatment to address post-SAH syndrome, for which there is no current treatment actually. With this, I would like to acknowledge the member of my team, Dr. Gavin Britz, which is the head of the Laboratory of Cerebrovascular Research, and he's leading the cerebrovascular field, with Dr. Eugene Golanov, who is a neuroscientist and a neurophysiologist in the laboratory, Dr. Nino Quirvelia, which is a research assistant, as well as Mrs. Chandra Sekaran, who is a research assistant also in our laboratory. I want to thank you for your time. I am Angelique Regnier-Golanov. I am an instructor in the Department of Neurosurgery. I conduct research in the cerebrovascular field. And I want to thank you for your time to listening to my presentation today.